Hi, hello everybody. Welcome back to another session of One Question a Day. The question that we are going to discuss is cementum, the general properties. The first point for that you need to mention is the mineralized cementum is the mineralized connective tissue covering the anatomical roots of the tooth from the cervical to the apical thirds. It begins at the cervical portion of along the cemento-enamel junction until the apical foramen. It is often thickest at the apex and thinnest along the cervical regions. The thickest ranges from 150 to 200 micrometers, whereas the thinnest is 20 to 50 micrometers. They are characteristically avascular and non-innervated, meaning they don't have blood vessels or nerves passing through them. So it is thickest at the apical region, about 150 to 200 micrometers, and thinnest at the cervical region, about 20 to 50 micrometers. They are avascular, non-innervated. They are hardness less harder than dentine with a slight yellow tinge color and very permeable, allowing metals and ions to pass through. They are believed to be about 45 to 50% having inorganic material, 50 to 55% having organic material, and some traces of water. The inorganic is mainly calcium and phosphate as hydroxyapatite crystals with traced element and has one of the highest fluoride content in the entire human body. This is very important point. The organic is mainly collagen. The type 1, the other traces present are the 3, 5, 6, and 12. The non-collagens include bone sialoproteins, osteonectin, osteopontin, and cementum attachment protein. This is unique because this is only found in the cementum attachment protein. The other proteoglycans such as chondroitin sulfate, heparin sulfate, growth factors are also found in traces. The types of cementum are traditionally classified as cell depending upon the cementum, the cells that form the cementoblast or the cementocyte seen. It is said to be cellular if there is cementocytes, acellular if there is absence of cementocytes. Based on fibers, they are classified as fibrillar when they have fibers, afibrillar when there is absence of fibers. The fibers are further classified as intrinsic fibers or extrinsic fibers. Intrinsic fibers when the fibers originate and end within the cementum. Extrinsics when either of them are outside. So we have important types of cementum: acellular, extrinsic, cellular, intrinsic, acellular, afibrillar. The other thing, acellular, intrinsic fiber, acellular mixed, cellular mixed. Cementoblasts appear as multinucleated cells and involved with cementum secretions. The classic difference between acellular and cellular cementum is acellular is first formed cementum. Cellular cementum is subsequent, formed later. Often seen only in single-rooted seed. Cellular cementum is missing in cellular. Cementocytes are absent in acellular. Cementocytes are present. Acellular begins from the cemento-enamel junction to apical third. Cellular, apical third and interradicular area. Acellular, it is widely believed that cementoblast derived from the remnants or parts of the HERS, whereas cellular cementum is believed to be a part of originate from dental follicles, particularly the dental sac region. The rate of formation of cementum, here it is slow and incremental lines are closer, whereas here rate of formation is fast and incremental lines are farther apart. The function of acellular cementage is anchorage, whereas here it is adaptation and repair. The incremental lines of cementum are referred to as incremental lines of Salter, which represents a rhythmic periodic deposition of cementum and appear as annular dark lines running parallel to root surface. They are highly mineralized areas with less collagen and more ground substances, seen both acellular and cellular cementum, but more prominent in cellular cementum. This is a paper of a different candidate to project to say how effectively diagrammatically the concept can be projected. Mineralized structure, outermost layer of anatomical root, avascular innervated, extends from CEJ to apex. This is how the nodes are being subsequently modified when the candidate feels that certain points are missed out. The hardness of cementum is less than of dentin, yellow in color, slightly lighter than lackluster, permeable to substances, chemical composition. Again, inorganic 45 to 50, organic 50 to 55 as calcium phosphate hydroxyapatite, highest fluoride content. Then collagen fibers, type 1 major, type 3 others, non-collagenous protein, proteoglycans, growth factors. Effectively, different way of the same content projected in a different way. Classification depending upon the presence or absence of cementum, acellular, cellular cementum, presence of fibers, intrinsic, extrinsic. The smaller size, the diameter of is about 1 to 2 micrometer, lie parallel. Extrinsic fibers are larger in size, about 5 to 7 micrometers on perpendicular, very important point. And formation of, and again, cementoblast. Cementoblasts, cementocytes are in cells incorporated into cellular cementums. They have processes entrapped in lacunae. They have canaliculi pointing towards, very important point, pointing towards the periodontal ligament. Deep to the cementum, they may transform into cementocytes. The symptoms of these transformations would include dilated endoplasmic reticulum, less number of mitochondria because they are quiescent cells, and cytoplasmic clumping with evidence of vesiculation. That brings an end to the discussion on the general properties of cementums if asked in an essay. Stay connected with this channel until we meet again with another question for one question a day. Happy learning incrementally.